Ah yes, the PlayStation 3, a console with some terrific highs and some terrible lows. But early on in its life cycle, it was used for a global science project, and that's what this lecture is on today. Welcome to Science After Hours. The PlayStation 3. The time the third console curse hit Sony and PlayStation. It was considerably overpriced at launch, and their main rival, Xbox, was doing something that some would argue are common features now in modern console video games at the very least. But we're not here to look at the mid-2000s gaming industry. Where is the science in that? Perhaps some data science, but I'm not a data scientist. Instead, we are going to look at how PS3s and how people at home were helping to fold proteins and push biological sciences forward. As I'm sure you are all thinking, where do proteins come from? I'm sure they don't just pop into existence when a cell divides, and you would of course be absolutely correct. This here is the central dogma of biology, where DNA becomes RNA, which is used to make proteins. DNA to RNA is transcription, where an RNA polymerase will run along a sequence of DNA, a gene, to produce a complementary strand of messenger RNA. This messenger RNA is made up of nucleotides, and each 3 nucleotides is a tricodon, and each tricodon corresponds to a specific amino acid. There are only 20 amino acids, so there is considerable redundancy between the codes, and by that you can see that multiple codons code for the same amino acid. You can also see that some of these codons code for things called a stop, which, as you might imagine, is the end of the amino acid sequence. And it is this amino acid sequence brought about by the mRNA sequence, which itself came from the sequence of the DNA, that is the primary structure of a protein. But to actually get the primary structure of a protein, translation needs to occur, which is transforming the RNA into the protein that we're going to talk about later in this video. So, and to do this, we need a special piece of biological machinery, the ribosome, which is a protein ribosomal rRNA complex machine that will run along the strand of mRNA, taking transfer RNAs, these special RNAs over here that have specific amino acids attached to them. This one here has a tryptophan attached, and it will enter the ribosome with each of the complementary tricodons on the mRNA and the tRNA matching up, and the ribosome will begin to attach the amino acids of each tRNA together. On average, a single protein contains approximately 300 amino acids, so therefore 300 of these tRNAs had to enter the ribosomal complex. But of course there is large variation in this. Some of the smallest proteins in the human body are estimated to be around 44 amino acids in length, whereas titin is the largest protein in the human body with 38,138 amino acids in its length. Here we have the primary structure. We've got 2 amino acids here. You can see that, so the R group is what designates what type of amino acid it is. They all have the same core structure. We'll talk about that in a minute, but you can see you've got amino acids attached to each other. Here's one, here's the start of another, here's the start of another. That is a snapshot of a part of the primary sequence. So this is the structure of the amino acid. You can see here a carbon with a double bond to an oxygen, and a nitrogen with a single bond to a hydrogen. A perfect recipe for a very strong type of intramolecular bond, the hydrogen bond. Unlike with the primary structure, we have different subtypes of secondary structure, and they are very much as a result of how and where these intramolecular forces occur. So they are therefore as a result of the primary structure. The first is the alpha helix, a right-handed helix where hydrogen bonds between that carbon double bond oxygen and the nitrogen-hydrogen single bond occur every 4 residues. You can see here, this is part of the same paper that showed you that primary structure. We also have beta sheets, which can be anti-parallel, where the 2 strands of protein go in opposite directions, and parallel beta sheets, when here the hydrogen bonds occur between the adjacent strands. Lastly, we also have turns and loops, which are the parts that connect the alpha helices and the beta sheets together. There are lots of types of turns and loops depending on the angle and the hydrogen bonds that hold it together, but think of it like this. Secondary structure is alpha helices, beta sheets connected together by a series of turns and loops. The tertiary structure is the 3-dimensional arrangement of all of the molecules that make up a protein. This is arguably what makes the protein that it is, in my opinion, as not all proteins have a quaternary structure, which we'll talk about in a brief second. The 3D nature of a protein is made up of a series of domains. Smaller proteins may be just a single domain, whereas larger proteins can have multiple domains, each one consisting of around 100 to 200 amino acid. Within each domain are motifs and folds, sequences that produce the spatial assembly of the secondary structure and thus produce the tertiary structure. So here you can see in this tertiary structure you've got a series of alpha helices, you've got some beta sheets in here, and this is all held together by intramolecular forces within those domains and motifs, holding it all into this protein structure, this 3D protein structure. We have already met a protein with a quaternary structure, that is hemoglobin. This is hemoglobin here. It's made up of 2 pairs of proteins, so 4 polypeptide chains. There's 2 here and there's 2 here. The quaternary structure is held together by the interactions between these different polypeptide chains by the intramolecular forces. There are currently several methods used to try and determine the complex 3D structure of a protein. There is X-ray crystallography, nuclear magnetic resonance spectroscopy, and electron microscopy. As someone who is a big fan of electron microscopy, let's take a little look at that last one first. So when people think of microscopy, you're probably thinking of shining a light on something, looking down the lens at your region of interest. Well, electron microscopy is not that dissimilar, except the major difference is instead of using light to view your sample, you are using a concentrated beam of electrons on a metal coated sample in a vacuum. This is obviously a lot more work than light microscopy, but the resolution and magnification one can get from transmission electron microscopy especially is thousands of times better than you will ever get with a light microscope, and this allows protein scientists to determine the structure of proteins, though it is not just as simple as firing your beam of electrons at your protein and then looking at it. The fundamentals of cryo-electron microscopy, the specialist form of electron microscopy that is used to try and determine the 3D structure of proteins, is not going to be the focus of this video, but basically your purified protein sample is flash frozen and then samples are exposed to electrons in the microscope, and the scatter produced is then recorded and reconstructed into your final protein structure. Nuclear magnetic resonance spectroscopy uses radio waves to determine the structure of proteins. Like before, you purify your protein sample and you place it in a strong magnetic field and blast some radio waves at it. The strong magnetic field will cause some of the atomic nuclei, especially hydrogen, to spin and resonate. The resonance can then be detected, and computational methods can calculate the atomic bonds based on the specific readings given. The major benefit of this method is that you can keep your protein samples in solution, whereas in electron microscopy and X-ray crystallography, that is most definitely not the case. So X-ray crystallography is probably the most common used method to determine the 3D structure of a protein in specialised laboratories around the planet. In this procedure, a protein is again purified, and as you might imagine, as the name suggests, it's then crystallised. An X-ray is fired through the crystal, and the pattern of diffraction is then analysed to determine the distribution of the electrons in the protein, and the resulting electron map and the electron density can then be used to interpret the location of each atom, and then you can use that to begin to calculate your 3D protein structure. Though not a protein, arguably the most famous piece of X-ray crystallography ever performed was done by Rosalind Franklin to confirm the structure of DNA, and aiding Watson and Crick in that monumental discovery. Why is it even important to know the 3D structure of proteins? Well, it's simple really. It can begin to help us determine how these proteins might function, and knowing how they function can help researchers devise drugs that can lodge into specific structures of the protein, or we can help synthesize new proteins that could be beneficial for us in making biofuels or degrading waste plastic, or for potential treatment opportunities. But there are major drawbacks of determining protein structures experimentally. As you might have gathered from just looking at the equipment, it can be extremely costly and it requires considerable knowledge and skill. And with things that require knowledge and skill, time is also important. We can't just have a quick look at a protein, figure out the structure, and then go on with our lives. But with the advances in computational power and the rise of artificial intelligence in the past couple of years, this has allowed estimations of protein structure to be calculated in silico or with computers. Arguably the most well-known AI software for calculating 3D structures is AlphaFold, a system developed by DeepMind, a subsidiary of Google. In 2018, it was first entered into CASP, the Critical Assessment of Structure Prediction, a community-wide experiment to determine and advance the state-of-the-art modelling in protein structure from amino acids, and it takes place every 2 years. In that 2018 CASP, it managed to beat the competition by a staggering 15% on all given protein structures, and at that time DeepMind used a bank of 128 machine learning processors taught on the roughly 170,000 known protein structures. In 2020, AlphaFold 2 was announced, and with some scientists describing it as the next step forward that could change everything in 3D protein structure predictions. Of course, it wasn't perfect, and in May of 2024, AlphaFold 3 was announced, and the version of this program can not only estimate the 3D structure of single amino acid chains, but can also estimate the structure of protein complexes with nucleic acids. CASP 16 is taking place this year, 2024, so the benefits of AlphaFold 3 are yet to be fully published, but based on their previous history, I'd be surprised if this is not another huge step forward. But it doesn't have to be done by AI. I know we're all worried about that Skynet-driven Terminator future that we could be living in soon enough. But this brings us right back to the PS3. There's no Skynet in the PS3. And an initiative allows us all to contribute to the calculation of 3D protein structures at home. And this is called Folding at Home, based out of the University of Stanford. This is a distributed computing project where everyone that downloads the Folding at Home client can contribute some of their computing power to the system. At the time of writing, Folding at Home is one of the fastest computing systems worldwide. At the height of the COVID-19 pandemic, for example, it had an operating speed of approximately 2.43 exaflops. To put that into a video game perspective, the Xbox Series X, the world's most powerful console as touted by Microsoft, has 12 teraflops of power. So 1 exaflop is a million teraflops, so 2.43 exaflops is 2.43 million teraflops, which is approximately 202,500 Xbox Series X's. So you can imagine, if you have an Xbox Series X, how much space almost 200,000 of these will take up. In 2007, the PS3 was more powerful than conventional CPUs, though slightly less powerful than GPUs at the time. As a result, it offered a suitable middle ground between speed and flexibility. The cell processor that powered the PS3 could run these protein folding simulations roughly 20 to 30 times faster than the standard PC at the time. Additionally, every single PS3 that had the client running and installed were identical, so that reduced the programming needs of the small folding at home team. The client was discontinued in 2012, but in those 5 years, 15 million PS3s had participated in the Folding at Home initiative, and in total donating more than 100 million computational hours. To date, Folding at Home has contributed to data to cancer work, to neurological disease work, infectious disease work, so that includes things like Alzheimer's, Parkinson's, hepatitis, and of course the aforementioned COVID-19. The best part? You can still download Folding at Home today. Obviously not on your PS3 and sadly not on any modern video game consoles, but if you have a PC or a Mac, you can go to the Folding at Home website and download the client. It's free, it's not some sort of Skynet spyware, it's just helping fold proteins. I'll put a link to it in the pinned comment below. It will run in the background of anything you're doing. You can adjust how much of your computational power you give to it, and then you can be part of a global science experiment whilst watching YouTube videos about science experiments. So I'm going to share my screen and let's see what my Mac is currently working on. So I am currently taking part in project 12,411. It's going to take roughly 10 hours and 21 minutes to complete. I'm only dedicating a small bit of power to this program right now because OBS is running, and that is a big sink in itself. So this is a 3D visualization of project 12,411, and Folding at Home also has a handy web client, and you can see here that project 12,411 is involved in influenza and hemagglutinin, which is a viral fusion protein that allows the virus to bind to host cells. So you, like me, can contribute to the determining of 3D protein structures of all sorts of proteins for all sorts of things. So I hope you give it a try, and I hope you've all learned something new today. Thank you for watching.